Hey everyone, good morning or afternoon. So this is just a quick recording about the lesson that we didn't get to finish on Tuesday because some of you guys asked for it. So it's not too bad. There's not too much left to pick up. So I'll just reiterate a few things. Remember that where we last left off was talking about lactose intolerance, and we're talking about people not being able to make the enzyme lactase, right? So they can't break down lactose. And we were moving you guys into this virtual lab. And in the virtual lab, there was a worksheet to be filled out that we gave you that is more that you'll finish after the lab. So when you press the link, right? For right now, we're going to ignore the team aspect of this because this is asynchronous, so you're doing it on your own most likely. So you'll start the activity, right? So you'll read it. Then it'll give you some good information about transcription. And transcription first. We'll talk about how DNA is being uncoiled or split. I usually just say split, but it's technically being uncoiled, right? We'll let it keep playing, right? You'll see RNA pops up a bit. We haven't given you the details that tell you all the different types of RNA, because RNA is incredibly important. But for right now, we're just gonna focus. We're just gonna go with just RNA for right now. Right. So see, once transcription occurs, the mRNA moves out, right? And then we go to translation. And translation is when basically it goes from DNA to RNA to proteins, and it goes through translation and transcription, right? And this is the little game. Start codons, right? So then you'll match the pairs, right? Just like we did last time. Remember the pairing rules. And this is an RNA, so remember RNA has a U instead of T, so AU should be right here, and you'll just complete this whole, you'll just cycle this whole thing and it'll stop. Okay? So once you do that, you'll answer the questions on the worksheet. Remember, when you're answering the questions on the worksheet, the first one, 3A, is about what this is trying to show you. So really think about like the definitions and the text box it's giving you. Make sure you read that. And then the second question is, what could you infer, meaning what could you get from this lab that you could say happens to people who are lactose intolerant? Like, what is going on in their genome? What is their body lacking? What does their body not have? Right? So that we're, really think about that and try to get that going. We'll go over that a little bit more in class, but I want you to take your best shot at it, knowing what we know about the genome. Okay, so we're gonna skip this because you guys are not gonna be in breakout rooms, right? And like I said on the virtual lab, how it showed the DNA uncoiling while there's DNA replication, right? So I think about it as a point where the DNA breaks, but it's uncoiling because remember it's a double helix structure, and it's starts separating, and both sides can operate as a base for a new strand. Because they're complementary, meaning that when a new side forms, it will match the existing base you already have, right? Because remember the nucleotide, the base rule, the nucleotide rule doesn't change, right? It's always A to T, it's always C to G, G to G to C, A to T, T to A, C to G. You put them in any order, it's the same thing: A to T, C to G, right? Or if you're talking about like RNA, A to U, right? T and U, you could switch out. Right, so as we're moving forward, like I said, do not forget the base pairing rules, and let's always remember that when it comes to DNA and RNA, order is really important because there is no breaking that rule, there is no rotating the two, or it's the order in which the A's, the T's, the C's, and the G's are in relates to what proteins are going to be shown and what traits are going to be shown, right? So if you change the DNA, down the road, you'll end up with a change in proteins. And if you have a change in proteins, you can get completely different things. Remember, it could be really physical. So you could be seen, think about like a dog's coat, so curly hair versus straight hair, or it could be chemical. Remember the cilantro. So like taste, it's chemical. You can't see it, but it's there, right? So I've kind of mentioned RNA a few times, and this one is really important to get because there's actually a lot of RNAs, and we're not going to go into extreme details because this is an asynchronous thing and you're not with us. And we can't watch this video because this is a brain pop, so I can't play the brain pop, and you guys don't all have accounts to it. So the things you need to know about RNA is that RNA is really similar to DNA, composition-wise, but they're not the same, right? So, for example, RNA is a single strand, right? So, like in the picture here, it is just one strand, and DNA is two. It's the double helix. It's the twisting, winding ladder, right? RNA has uracil, while DNA has thymine. So it's the T versus the U. I found these videos, which I think you guys should be able to play. I'll put the links right underneath this video on the Google Slides. The first one is some pretty basic differences between the two, so that way you can kind of see why the difference only 2 minutes. And I'll also put this other one, DNA and RNA updated. I like this one a lot. This one is a little bit longer. It's a little more detailed, and if you guys really want to challenge yourselves, I recommend you watch the full thing. And even if you don't fully get it, watching it through once or twice will help you familiarize with RNA, because there's a few of them. There's messenger RNA, ribosomal, transfer. So there's a few, and they're all really important for what they do when we talk about expressing traits from DNA. They're all key steps. What is the next step? Right? So how do we get RNA, right? Because remember on the model, the Google Slide models we did, we had DNA going into RNA, going to proteins, going to traits. How does that happen? Well, it goes through DNA going into RNA, goes through the process called transcription. That's where the DNA is split, right? It's uncoiled, it's broke apart, and an RNA is made from one side of it, right? So there's an enzyme attached to it, and it starts to make an RNA, right? That happens through transcription. And then it sounds really familiar, but it's not the same word, right? That RNA will undergo translation. Undergoing translation, it's being read and matched like in the virtual lab, and it's creating proteins. It's creating like a chain of proteins. And proteins will connect to each other and be wound together, and depending on what the order is and what the sequence and the section that we're talking about, you'll get brown hair, blonde hair, red hair. Maybe it's about height. Maybe it's about eye color, depending on what the location for that gene is, right? So let's try to remember that. So we know it goes DNA, RNA, protein, traits. So let's put in the two more. So it goes DNA through transcription to RNA, from RNA through translation to proteins, and you'll get traits. All right, so it's really important to remember transcription and translation, because those are going to be key factors as we go down this road, right? And I put this slide here because I think it's a little easier for that, because they do really matter. They're incredibly important. So DNA holds all your genetic information, right? And it has to undergo transcription to go into RNA, because DNA by itself will not get you your traits. It will not express it. It needs to undergo steps. Things need to happen to it in order for it to be read, right? For it to be turned into something that happens to an organism's body, anatomy, structure, right? So it's going to undergo transcription, right? So it's like writing it down, and it becomes RNA, right? And then from RNA, you need one more step before it can become a trait, proteins to start going to traits. It needs to go to translation. So it's like DNA gets written down into RNA through transcription, and then RNA gets translated into proteins. So now we're no longer talking about A's and G's and T's and C's and U's. We're not talking about these bases anymore. Now we're talking about proteins. So you can think about it like it's another language. So it's being translated into another language. So RNA is being translated into proteins. Okay? And then proteins will give you traits. Curly hair, eye color, and all that other stuff. There's this, I think we played it for 7th period. Or no, maybe we only played it for, I think we only played it for 5th period maybe. But the link will be here. Oh, if it wants to open. Okay, that's not working. I will make sure that link works before you guys get it, so that way it's okay. But it's another video that talks about transcription and translation. The other 2 YouTube videos will cover that too, and with the worksheet and stuff, you'll have a good familiarity with it. So lastly, to cover this up, is let's remember some important takeaways, right? Let's remember DNA is double-stranded. It's a double helix, right? A, C's, G's, and T's. Base pairing rule is always the same. A to T, C to G's. RNA is single-stranded. Follows the same base pairing rules, except it has a U, not a T. So it's A to U and C to G, right? Genomic materials all stored in an organism's genome. And remember, we knew the original flow. It's DNA to RNA to proteins. And now we're saying DNA, transcription to RNA, so it's like being written down. And then RNA is being translated, right, into proteins. So it's being like, it's going from the bases, the A's, the T's, the C's, and the G's to proteins. So let's try to remember that's how that process works. Okay? And then lastly, I don't have it pulled up right now, but the academic prep for the asynchronous lesson is going to go into, it's an individual assignment. It's an individual assignment where you're going to write down your opinions on the topic of designer babies and what you think about it so far, whether you're for or against it, and then you're going to write down some of your own arguments for you on your side. So it's 2 to 3. So if you're for it, write 2 to 3 arguments that you would tell your group mates as to why you think this concept's a good thing. Or if you're against it, 2 to 3 arguments why you think it's a bad thing. Because you guys are going to sit down together in a group soon and talk about it and come to like a group decision for you guys' project. Any last questions? Someone asked me what stance means. So stance is like your position, what you're trying to get across, your claim. So if it says what is your stance, it is what is your position, what is your claim. And I think that's about it. If you guys have any help, need anything, always email us. We have office hours today at 11 to 12:30. But other than that, you guys are all set. All right, have a good weekend, and I will see you guys on Monday. Yeah, on Monday. All right, bye guys. Have a good one.